All right. Hello, everyone. Thank you so much for registering and attending the Microbiome Amplicon Sequence Analysis Pipeline and R workshop at the Tri-Societies meeting this fall. My name is Mara Cloutier, and I'm a PhD candidate in soil science and biogeochemistry at Penn State University. I put my email address here in case any of you need to reach out to me and ask any questions while you're going through the workshop, or if you have any questions, in the near future about any of this, any of the information that I've presented in the workshop today. I'm really sad that I can't be there giving this to you all in person because I was really looking forward to being able to interact with everyone. But I still think I've put together a pretty good virtual workshop for you, and I hope that you all learned something from today's presentation and in the tutorial in general. So why me? Why am I doing this? Well, I can tell you that I've got 3 publications, first author publications, that have used Amplicon sequencing, and mostly all in soil science and biogeochemistry related fields. And through my time at Penn State, I've really found that the whole idea of teaching yourself is really like, oh, you have to teach yourself. It's not really, there's not many resources for people to go and get help and to work through these pipelines. And so I started the data analysis working group for the Microbiome Center at Penn State, where we take a professor's Amplicon data set that they have not been able to analyze or just don't have the skills to analyze, and we analyze it for them. And while we're going through the analysis ourselves as like the 4 group leaders, we also work on, breaking that analysis into workshops that we give to graduate students and postdocs. And here's a photo of our group on the left here, where we're working through a data set from 2 semesters ago. Currently, the leaders of DOG, including myself, we're working on writing 2 publications with the professors whose data we've been working with. So hopefully those will be out, relatively soon. Um, for the purpose of this workshop, I decided to skip all of the background information on, what's amplicon sequencing and, what, how you should collect samples or things like ASVs versus OTUs. And I just decided to go straight into basically the tutorial. But if you want some more information about like ASVs versus OTUs, then I put here 2 links to 2 papers that go into detail about comparing the 2 methods and what really showed the best results for what they were looking at. So you can kind of decide for yourself which one is best. I will say though that the pipeline that we use today is for ASVs, and it cannot be um, created for OTUs. So hopefully you'll want to be using ASVs after today, because it's a really easy pipeline that the people who created data 2 started for us all. It's really user-friendly. It's all in R, which makes it even more helpful and easier to use. And hopefully you'll start to see why I like it so much when we go through the tutorial. So the first thing that you need to do is you need to go to GitHub. And specifically in your web address up here, you should type in github.com/Mara Cache/Tri-Societies-Amplicon-workshop. Now, this is where I've got all the files that you'll need for the workshop today, as well as a few instructions that you'll need to get through the tutorial. And so once you get to this website, then you'll see there's some files here. There's an amplicon-workshop.r file. That is going to be the R script that we're going to be working through. And then there's a metadata.txt file. And that file has all of the information that we need about the sample. So it has what block they came from, what sorghum line they're from, what are some root chemical concentrations that associated with each of the samples that we're looking at that will use in our analyses. And then there's a bunch of fastq.gz files. And now these are all of our Amplicon sequences here. And then below that, I have just a little bit about the workshop, and that we'll be using data 2 to process and filter the Amplicon sequencing reads. And then we're going to create a phylogenetic tree and then perform some downstream analyses such as creating bar plots of taxa, alpha diversity, beta diversity, and differential abundance analyses. There's a few other things mixed in there, but that's the main things that we'll be doing. And then as well as just a few instructions on the 4 things that you need to do to really get started. The first thing is to download the GZ files, the metadata file, and the Amplicon workshop script. Now, the easiest way to do that is to click on code and then hit download zip. And this should download all the files that are in this GitHub repository. Okay, so let's just say we'll do that. Then while that's downloading, you need to travel over to the Zenodo website and you need to download the reference database. So this is a 16S reference database, Silva database, that we will use to assign taxonomy to our Amplicon sequences. Once you go to the Zenodo site, then you'll notice that there's several different Silva databases here. And the one that we need for today is the Silva NR99 V138 train set. So you can just say, oh, I'm going to download that. And then that download will begin and travel back to the GitHub site. And the next 2 steps are pretty easy. They're going to be to open RStudio and the R script and then just start working from the R script. I also put in a few disclaimers here that you should be aware of. So the first one is that if you're working from an older R version or an older RStudio version, then there may be some initial issues with package installation. And the easiest, best way to deal with that is to just update your R in your RStudio and then try and reinstall these packages. Because sometimes there's just compatibility issues where you can't even get the R package to install on older versions. And also I think you should know that, once you go through this tutorial and you have this code that you can pull from for your own data, it's important to think about the questions and the hypotheses that you have for the data set that you're working with. The methods and analyses here may only just be a good starting point and may not necessarily be the best methods for your specific data, but then again, maybe they are the best methods for analyzing your data, but you have to be the judge of that. And know that these are not the only ways that you can analyze the data. There's lots of other, pipelines, tutorials, everything to pull from where you can try and figure out what is really best for you. Okay, let's check out our downloads file or folder and see what we got. So we have the Silva trainset.fa, which is great. And then we have the Tri-Society's Amplicon workshop, which is also fantastic. That's actually this one for me. So we can see we got the Amplicon workshop.r metadata and then a bunch of FASTQ files. Now, once we have everything downloaded, we can start in R. And I am actually going to say, R, I want to start a new session. But I'm going to keep my other sessions open because I don't want to lose where I was. Okay. And let's say that we want to open our R script. So we would say file, open file, and then we're going to travel to our workshop area and we're going to say we want to open this Amplicon workshop.r file. And then you'll see it pops up in our RStudio. And I have a bunch of steps. I have everything blocked off in steps of what we'll go through today. And the first step is to install packages. So we'll start with line 2 here, and we'll just work our way down, and hopefully you don't have any issues. Keep in mind that if you do use like the analyses and kind of like this pipeline, then these R packages would need to be cited in any publications that you're using. Okay, so it says update all, some, or none of these older packages, and I don't think I need to update them, so I'm just going to put an N here. And then we'll just keep running through. We'll run DevTools to install corncob. That's fine. It's just saying that I already have it installed and I can reinstall it if I use force, but I don't need to do that. And we'll say, okay, well, we're going to install some more packages. Do you want to start R, restart R? I would say yes. Sometimes this is really important because sometimes if you're installing a package in R and it asks if you want to restart R, if you don't restart R, the package that you installed won't actually be able to run. Yes, and sometimes this can take quite a while to get all the packages installed. And then we're going to run this cool little function, iPack, which is just going to make package installation a little easier for us. And then it's actually going to tell us whether or not it was able to install the packages that we tried to install. So at this step, what you should be seeing is that under package data 2, you see true. And that's the same case for all of the packages listed here, is that they're all true. If one of them is false, then that means that it was unable to install that package, and you may have to just manually go in and try and install it, which would look something like this. You would just say, okay, well, let's say it's biostrings that didn't install. And then you can look up here and you could say, okay, biostrings is here, so that means it's within this BioC manager install. So let's copy that and we'll put it here and we'll say, okay, we need to try and reinstall biostrings. Okay, and then you would just run that. Now R has this funny little thing about it where you have to install a package, but in order to use that package, you have to bring it into your library, which I always thought was kind of confusing and I didn't quite understand it. So to bring it into the library, you just say, okay, library biostrings. And then there you go, you can start using that. Now, if it's not a BioC Manager package that you had the issue with, then hopefully you can install it just saying install packages, let's say vegan. And then you would say, okay, you're going to run that. Let's not restart R on this one because I don't really think it matters for vegan. Okay, so it installed, and then you come here and you say, oh, I want to put it into my library. And voila, it's in your library. Assuming that all of your packages installed, we can move on and actually start the tutorial. Now, the first thing that you're going to do here is you're going to set your working directory. You can do that on the command line, but I actually have found that it's much easier to set your working directory by going to Session, Set Working Directory, and then choose Directory. And then you just navigate to wherever the folder is. So for us, it's this Tri-Societies Amplicon workshop. You say open, and then you set your working directory. If your working directory is not downloads Tri-Society's Amplicon workshop, then what you're going to need to do is copy where your working directory is, and then overwrite it here. Okay, because what we need to do is we need to tell R that we want to start pulling files from the Amplicon's workshop folder, right? And then we can say, okay, well, now let's see what files are actually in that folder. And we can see, well, there's this R script, and then metadata file, and then there's a bunch of fastq.gz files, which is great. That's exactly what we want to see. After we have our working directory set, and we're going to be pulling files from the correct location, then we just need to do some sorting of the files. So we're going to run this fsam here, and then the rsam. Basically, it's just saying, okay, well, if the file name has r1_001.fastq.gz, then we know that that's a forward read, right? Because all our R1s are forward reads. And then if it's an R2, then that is a reverse read or a reverse sample, okay? And then we can actually extract sample names from what we had just created with fsam, and then we can look at those sample names. So now, instead of a sample name being R3-5A underscore S215 L001 R1 yada yada yada, now our sample names actually make sense because they're the sample names that I gave to the sample, which is R3-5A. That means that it's the third replication of the near isogenic line 5 for our sorghum bicolor that we're looking at, okay? So what we've done so far is we've basically told R, okay, these are the files that we're going to be working with, and these are how we want to name the files. But we haven't actually started to like look at the quality of what's in those files, right? Like we haven't started looking at the actual sequences yet, and that's what we're going to do in step 4. So here we're going to plot the quality profile of samples 1 through 4, okay? We can easily change this to be like, 11 through 14 or something. And we just say, oh, sorry, we don't have that many samples. No, we can't do that. We can say 5 through 9. How about that? Because we have 9 samples. And it does take a minute to actually create these quality profiles, I think, because there's just so much information that's jammed into what we're looking at. And throughout this tutorial, I have a bunch of comments on how to interpret things or what to do next or what happens at certain stages in the code. And so, if you're ever stuck at something, you can always just see, is there a comment there that I should be looking at? Maybe that'll be helpful. Okay, so with these plot quality profiles, there's a lot of information included in this. So let's just zoom in here. Okay, and we've got 1, 2, 3, 4, 5 total samples that we're looking at. Okay, and then it tells you how many reads there are in each sample. So in this specific sample, R248, we've got 51,000 plus sequences, and then over in this last one, we got 49,000. The grayscale represents the frequency of each quality score at each base pair. So let's look here. Let's make it make sense, okay? So we've got quality score on the y-axis, and then we have the base pair length here on the x-axis. So like when we're looking over here at the far right on the plots, that's close to 250 base pairs in length, which is what we expect because what we did for this project was we did 250 by 250 base pair sequencing. So each read should be 250 base pairs in length. Okay. And so the grayscale represents is a heat map of the frequency of each quality score at each base pair through here. Okay. So what you really want to see is that the grayscale is greater when we get, above 30, right? So seeing the quality score between 30 and 40 is most ideal. Okay. And then, which is really what we see here, right? Like up here, close to 40, where the orange and the green lines are. So the green line represents the mean quality score at each base pair. So we can see at the beginning of the reads, it's high, 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 and then it kind of just starts to slowly become just a little bit less, have a little bit lower quality score, which is really typical. Right, is that the further you sequence, like the more base pairs you sequence, the quality score just decreases just a little bit. And the orange line represents the quartiles of the quality scores at each base pair. And the red line here down towards the bottom represents the number of reads that extend to at least that position. Now this is really not that important for Illumina reads because Illumina reads are so precise where you end up with, all of your sequences are 250 base pairs in length. This becomes a little bit more important if you're doing like pyrosequencing where they can vary in length by many base pairs. The biggest things that you want to take away from here is you want to make sure that the quality is fairly high across the whole 250 base pairs. And there's ways that you can account for lower quality scores in your sequences. I should also mention that these are the forward reads, right? So let's look at what happens when we do the same sort of analysis, but for the reverse reads here. Okay. So we see a pretty much similar trend, right? Where like the quality score is definitely between 30 and 40, and it's the mean quality score goes down just a little bit across the sequencing length. But overall, these are really high quality sequences. And I have seen some projects that did not have very high quality sequences, especially for the reverse reads. And if you see that for the reverse reads, don't be too alarmed because it actually has a lot more to do with the Illumina chemistry that they use for sequencing, where what they do is they sequence the forward reads first and then the reverse reads. And so by the time it gets back to the reverse reads, then the reaction isn't quite as efficient, so the quality scores tend to drop. And after we've kind of looked at the quality and we've kind of have an idea about like if we what we should filter and if we need to trim our sequences, I usually like to trim off at least like 10 base pairs from the end of my sequences. So if they're 250, then I at least like to trim off to 240. And we'll see how I did that in just a second. But first, we need to set R up for the filtering step. So we just need to run a few lines of code here before we get to this filter and trim step. I have a lot of information here about filtering and trimming. And let's see, this can be, there's a lot of different commands included in this function that really can help to make sure that you're doing the best for the sequences and the quality of your sequences that you're working with, right? So the first 4 things that you have here are basically just input files, right? So you have a set of files that are going to be your filtered forward reads and then your filtered reverse reads. And then we have this trunc length command. Now this command is where you're going to be telling it to actually truncate the length of the sequence at whatever base pair. Like I said, I at least like to cut off a few, even if they're pretty high quality. And so for the forward reads, we're going to cut it to 240. Now, as I had also previously said, that the reverse reads tend to be poorer quality than the forward reads. So even though that's not necessarily the case for this data set, I still just set the truncate length to be 160 for the reverse. Now, you don't want to truncate too much from your forward and your reverse reads, because you still want them to overlap enough so that you can actually merge the two, right? Because the whole idea of sequencing forward and reverse reads is that you can put them together and create a higher quality, nicer, and longer contig, right? If you're using the 515F and the 806R primers, then the information on this website, this Illumina website, can be really helpful in understanding how much is okay to cut off and still have some overlap. Okay. And there's other things included in this step, which is that max N. So for this, I always set it to 0, but again, you need to be the judge of that for your own data set, where N is the number of ambiguous base pairs that you will allow. I didn't feel setting M means that all ambiguous base pairs will be removed, okay? And for max EE, this is the maximum expected errors that you will allow until the read is going to be discarded. So if it has 3 maximum expected errors, the sequence does, then it will just remove it from the data set. 2 is the default, and you need, so the first 2 here is the maximum number of expected errors for the forward reads, and then the second 2 is the maximum number of expected errors for the reverse reads. And then there's this trunc Q command here, which is also set to 2, and that is again the default, where it truncates the reads where the quality score drops less than or equal to the number specified. And then I always like to compress so that it'll be, instead of a .fastq file, it will compress it to a .gz file, which just takes up less space on your computer. And then multi-threading, I always set to true as well, which just speeds up the process. And if you're on a Windows, then you need to make sure that, well, you need to set it to false if you're on Windows, okay? So let's just run that command and you'll see, so it's creating the output directory. So it's going to be a filtered folder within the Tri-Society's Amplicon workshop folder, okay? And it's just going to throw the filtered reads in that folder. This does take a few minutes to run, so don't be alarmed if it is just kind of sitting there for like 10 minutes or something. And then you can inspect how many reads were filtered out. And this is really important. You want to know that, okay? So what you should see if you run head out is that it'll give you a truncated list of your samples. So here, instead of showing all 9 samples, it's showing 6 of the samples, okay? And the first column is the reads in. So this is how many reads we started with per sample. And then the second column is the number of reads that we have after the filtering step. Now, if your reads are poor quality reads, you may see that you filter out half your reads at this step using the function in the way that I have here. At which point you would want to go back and you would really probably try to try a less conservative approach. So you might try setting max EE to 3 and trunc Q to 3, or playing around with your truncate length, or allowing for ambiguous base pairs, right? There's options here for you to play around with, but you need to understand that you're kind of walking a fine line where you want to be working with quality data, but you still want enough of that quality data to accurately represent your sample. Then let's say you're happy with the output from this filter and trim step, and you can move on to actually assessing errors. Right? So samples, sequences have errors in them, and data was built to actually model the errors that is expected within a data set. And we'll see what that looks like in just a second. So let's run this first line here where we're learning the errors for the forward reads. So what it really does is it tries to distinguish errors from the true exact sequence variants, right? So it's trying to say, okay, well, these sequences have shown up so many times, they must be biologically accurate and without error. And then this, like maybe one sequence has one base pair that's different between the sequence and then this whole other group of 100% identical sequences. So it might actually say, okay, well, that base pair is likely to be an error. So what should it actually be? You know, it says it's an A, but all these other sequences have it as a T or a C or a G. So what's the likelihood of that error actually occurring? And for this pipeline, we need to learn the errors for both the forward reads and for the reverse reads. We'll run this on the reverse reads after it finishes with the forward reads. Hopefully you all have signed up for either like the Wednesday session for the boardroom or the Friday session, and we'll be able to come in and ask any questions that you have while you're, you know, either going through this tutorial or you know, any really Amplicon sequence question that you have. The sessions are set for 2 hours, so you can, you know, come in for the whole time, or you can just pop in. But I'll be there the whole time for both the sessions on Wednesday and on Friday. And these learning error modeling steps do also take some time to run, you know, because it's essentially it's building a model, you know, of what your expected errors are. And hopefully that too, that the modeled errors are representative of the errors in your sequences. And there's a way when we move down to this plot errors, we'll actually see how well the errors modeled against the sequences that are in your samples. And so you'll notice too, so it says, okay, well, it used this many number of total bases and this many reads from 9 samples to learn the error rates. So what it really does is it takes like a subset of your sequences and models the error rates. Okay, and then we can move on. We can run it for the reverse reads. Great, so we have modeled our errors, and now we can actually look at what those models look like in comparison to our samples. And so what you're looking at here is, is the consensus quality score on the x-axis and the error frequency log 10 on the y-axis. And then you'll see that there's A to A, right? And then there's, okay, well, if it's an A going to a C, or an A to a G, A to a T, or a G to an A, or a T to an A, and so forth. Now what you should see here is that your samples actually track well, your samples are the circles. You should see that your samples track well with the black line, right? Yes, and which is normally the case. Unfortunately, the people who created data don't provide too much information on how to really assess what something looks like when your samples don't track well with the black line. So let's hope that yours do. And for me, I would say, let's look at this again. Is it, no, can we zoom in on this? Oh, wait, no, no, it's back. Let's trying to get to the image again. For this purpose of this tutorial, I'd say that generally the samples track pretty well with the black line, and we don't need to be too worried about poor error modeling. After error modeling, then we dereplicate our samples. So basically what we're saying is if there's 10 samples or 10 sequences that all have the exact same sequence, so they're 100% identical across the 250 base pairs, that we're just going to keep one. Now the reason to do this is because it really helps with downstream analyses. It speeds it up. Okay, and then we'll bring back in, so we're going to store that information, right? So about like, okay, well, there was 250 sequences that were all identical. And so, and then this is the sequence. So we'll store that so we can come back and then add it back in. Right, so you'll see here it goes through each sample one by one. Right, so in the last sample here it says encountered 18,466 unique sequences from 45,000 total sequences. Right? And then we'll do that again for the reverse reads. And then we just need to store some sample names. And then we can actually perform the data step of the data to pipeline. So we'll get the forward data step going. And basically it's taking your dereplicated samples and your modeled errors. Okay? And it's going to go through every sequence in your sample and it's going to remove sequences that are actually like sequence errors or have sequencing errors, okay? And like you'll see, for sample one, there was 5,000 or 50,000 reads in 20,000 unique sequences, right? So it's just going to go through each sample one by one and actually infer sequence variants. So what is a true biological sequence? And obviously, depending on how many samples you have, you can scale the amount of time it's going to take to go through the pipelines and all the steps. Which is why for this, I decided to just work with 9 samples instead of say like 50. Because if we were working with 50, it would take us a lot longer to get through the pipeline. And if we're almost done here, and we can kind of check and see like what's going on, what happened for the forward reads and the reverse reads by doing something like, okay, well, tell me what happened with sample 3. Right? So it'll say, okay, in sample 3 there was 654 sequence variants that were inferred from 12,000 input unique sequences. And then, if you really wanted to, you could go through all the other samples, but I wouldn't necessarily recommend doing that. And then in the reverse read for sample, well, let's just change this to sample 3 too. So, okay, well, there was 571 sequence variants that were inferred from 9,000 input sequences. So now that we have our sequence variants that were just calculated using data, we can go back and put together our dereplicated samples. And we're also going to pair the reads here too. So it's going to say, okay, well, this forward read goes with this reverse read and so forth. So you'll see this is probably for the last sample, sample 9, that there was 37,000 paired reads and 524 unique pairings successfully merged out of, 41,000 paired reads and 1830 pairings, so forth. So this is just giving you an update of what happened in each of the samples. So we can say, okay, well, let's look at that a little bit differently. And we can say, all right, so for sample 1, there was one sample that was found, or one sequence in sample 1 that was found 7,000 times. And there was a nucleotide match. So this is going to be the overlapping region between the forward and reverse reads of 147. Okay, so like I had previously said in that filter and trim step, if you truncate too much, you're not going to have enough to overlap, and you want quite a few base pairs there to overlap, so you know that those two reads go together, right? So this is just saying, well, even though we truncated 10 off our forward and we truncated to like 160 on our reverse, there was still 147 base pair overlap, which is pretty good. The number of ambiguous base pairs is 0. The number of indels is 0. And it goes through basically, like, if you wanted to, you can inspect every sequence for every sample, which is a little bit too much for this tutorial. It may not really be worth your time. It may be important for certain samples and just certain situations. It's very situationally dependent. So now what we can do is we can actually say, okay, well, let's look at the distribution of the sequence lengths, and also how many total sequence variants we have across the samples. So that is what this is saying. Is that, okay, well, in our sequence table, this file, we have 9 samples. Of those 9 samples, we have 2,205 total ASVs. So the Amplicon sequence variances, there's 2,205. And we can look at the sequence lengths here, and we can say, okay, so there are 15 sequences that are 240 base pairs in length. And then there's 2,000 sequences that are 253 base pairs in length. And then we have, like one for 260 base pair, one for 288, and then we have one for 337. Depending on this distribution, you may or may not want to truncate some of these sequences. So let's say, like, that I don't really care about reads that are less than 240 or greater than 260 in base pair length, because it just doesn't seem like it may be a true biological sequence. So we can say, okay, well, we're gonna truncate sequences that don't fall within that window. And then you'll see here that we now have the seqtab2 file, which has sequences that are anywhere from 240 to 260 in length. Then we move on to step 10, which is where we're going to remove chimeric sequences. And for some reason, Data2 has decided they're actually going to call it bimeras. And so this is the code to do that. So we'll go ahead and get this started here. And it says, okay, well, it identified 126 bimeras out of 2,203 input sequences. So for those of you who aren't familiar with chimeric sequences, it's basically when 2 reads are sequenced as one, but they actually shouldn't be one sequence. That's something that happens all the time in this high throughput sequencing sort of methodology. And we can actually look at, say, okay, well, there's 126. Is that really important? Is that a lot of our sequences? Is that a few of our sequences? Just get an better idea of how well the like further idea of quality of the sequencing. So we can run this here and say, oh, not that one. And we can run this one here. Percent of sequences that are non-chimeric. So essentially, 95.7% of our ASVs are non-chimeric, which is a really good percentage. So that means that less than 5% are chimeric. 5 years ago, when I first started doing Amplicon sequence analysis, the number of chimeric reads in papers was like 30%, which may still be true depending on whether or not you're sequencing bacteria or fungi. I think chimeric reads for fungi tend to be a little greater of your percentages. So now we can kind of look at, okay, well, how many reads have we lost at each step? So we've done filtering and trimming, we've done merging, a bunch of different steps here. So let's take a look at what has actually happened to all of our sequences. So again, this just gives you an abbreviated list of the samples. So we've got 6 here, and we've got, okay, well, there was this many sequences that we originally started in the input. And then after the filtering and trimming, we were left with this many sequences. Denoising resulted in this many sequences. Merging, so putting the forward and reverse reads together, resulted in this many sequences still being kept, and then the number of sequences that were removed when we cut sequences that were above or below a certain sequence length. So like removing the sample or the sequence that was 337 base pairs in length. And then all of the sequences that were non-chimeric. This is really important for you to pay attention to. So if at any step in the process you lose a lot of your sequences, you should go back and to that specific step and decide whether or not you want to tweak some parameters, right? You might want to, if you're losing a lot, then you're going to want to be less conservative. So you'll allow for sequences with more expected reads to pass, or sequences that, like if you're merging forward and reverse reads, maybe you'll allow for there to be one base pair different between the overlapping region, and so forth. The last major step of the data pipeline is to assign taxonomy to our sequences. So we've filtered, we trimmed, we've processed, and now we can assign taxonomy to those 16S sequences. And we've already downloaded the Silva database, so we have that and we can use that for taxonomic assignment. Okay, perfect. So we have taxonomy assigned, and then we can just take a look at that to make sure that everything looks copacetic. So we have our sequences here, which, is great. And then we have, okay, well, we can just scroll up here. How about that? Say, okay, kingdom, the sequence belongs to bacteria, phylum, sequence belongs to proteobacteria, classes is gamma proteobacteria, order pseudomonadales, and so forth, which all looks great. And now we can actually start to build what we call a phyloseq object, which is a fantastic resource that is basically one object that includes the samples, so the sequences and their abundances, the taxonomy of those sequences, and then the metadata associated with the samples. So before we bring in this metadata file, I want to actually just take a moment to show you what it's supposed to look like, because for me, it took me a long time to figure out how to best format a metadata file and what sort of information should really be included. So let's take a look at the Excel version of this metadata table. Okay, so we have variables on the first row. The first variable is the sample. So these are our samples here. And then this label, this is something that is specific to, the study itself. So basically like N321 is the near isogenic line of sorghum that we were using, and N336 and N334, right? And we were using rep 1, 2, and 3. So this is like R1, R2, R3, right? And we have here the genotypes. So we're using 5 total genotypes. I've chosen samples from 3 of them, so 3, 4, and 5. And then I have the line here, and then I have some other information that from each of these rhizospheres, we use the roots, and we actually quantified certain things in the roots themselves. So we looked at total phenolics, this DPPH, which is an antioxidant assay, and then luteolinidin, I just called LUTs because it's easier, and then flavonoids, also I called flavs because again it's just easier, and then I have the concentrations here. So this is kind of what your metadata file should look like if you're going to bring it into R. Somewhat formatted similarly, obviously, like your column names and what's in your columns are going to be different because you're working with a completely different data set, but this is the format. Okay. So let's go back to R, and we can say, okay, we're ready to read in our metadata file. And the headers, so like that first row where we have the sample variables, so like sample name, lutes, flavs, etc., we want that to actually be the column names. Okay, we don't want it to be just a row of variables. We want them to be the column names. So we're going to say, okay, header is equal to true. And then we want the row names to be the actual sample names. So we're going to set row names equal to 1. So we import that, and we can say, okay, well, let's just look at it and make sure it all imported right. So we have the sample names as the row names, which is what we wanted, and then we have the variables as the column names, which is, again, exactly what we wanted. So that went well. We're going to convert the sequence table file that we have, the seqtab.non-chimeric sequencing table. We're going to convert that to an OTU table, which is just something, within R that we need to do. So we're going to say, okay, well, we're just going to convert it to an ASV table. And then from that, we can say, all right, we're ready to build a phyloseq object. So we'll say, okay, we're going to make the phyloseq object with our ASV table, with the taxonomy table, and with the sample metadata. All right, let's do it. Perfect. It all worked out well, which is great. Sometimes I have an issue with that. The next step, you may or may not want to make a phylogenetic tree. The easiest way that I have found to do that in R is to actually follow the pipeline from the paper here that I have. It's from Benjamin Callahan's group, where they have a huge pipeline on how to do bioinformatics with data 2 and R. And part of that includes how to build a phylogenetic tree, which I found incredibly, incredibly helpful. So we're going to go through that pipeline. We're just going to say, okay, well, we're going to use our sequences, and then we're going to make a multiple sequence alignment using this align seqs command here. Oh, please keep in mind that if you did ITS sequencing, it is not necessarily advisable to perform to make a phylogenetic tree with those sequences. if you're using like 18S sequences, your phylogenetic tree is going to be much more reliable, but there's just so little conservation in that ITS, in those ITS sequences, that it's really hard to build a good quality phylogenetic tree. All right, so we have our multiple sequence alignment done, and then we can use that. We're going to make a neighbor joining tree. All right, and then I have to let you know that these two steps, this fit GTR, they do take a little while. Well, the first step doesn't take that long, but the second step definitely takes a little while to actually, so what it does is it takes the neighbor joining tree and it makes a generalized time reversible with gamma rate variation maximum likelihood tree. And this step does take a little while, so just be prepared for that. And so then we can detach the package Vanguard because it actually takes up quite a bit of space, so we'll do that. And I'll say, okay, so we made our phylogenetic tree. Great. Now, what we can do is we can add that into our phyloseq objects, which we will do. And then I'm just going to duplicate the phyloseq object, which is this PS, and I'm going to make a PS1. It's the exact same thing. I just always like to keep the original and then work on the copied or the duplicated version. Right, so we'll say, okay, we're gonna duplicate the phyloseq object, and then what I should really show you is what it looks like. We say, okay, we want to look at the OTU table of the phyloseq object, say of PS1. All right, great. Oh, well, we see just sequences. And what we should really change is we should change those sequences out for ASVs. And the following lines of code will do that for us. Okay, great. So instead of seeing, sequences that are all 250 base pairs in length, what we see now is those have been replaced with ASV numbers. So it just will make the downstream analyses run just a little bit faster and a little bit easier to interpret. Okay, and then we can say, all right, we just make sure that everything is there that we need. Let's say, all right, we're gonna say, look at the taxonomy table and we can see, if we scroll up here, we can see, okay, we got a, kingdom, phylum, class, order, family, and then genus, right? So that's what we should see. And then if we can look at our sample data and we can say, all right, great, we've got genotypes, lines, blocks, total phenolics, all that information that we also want to be using. Okay. Now, one thing that has been confusing to me in the past is that Silva is based on, the Silva object that we use for our reference database is 16S sequences. But there are also some eukaryotic sequences in there as well. So it's always a good idea to run something like this next line of code where we subset and we say, okay, what we want to do is from our phyloseq object, we want to keep all the sequences that were assigned to the kingdom of either bacteria or archaea. Okay? And we're going to put all that information into this new phyloseq object, PS2. Great. And then sometimes what you'll see is like, 5 sequences have been removed, or here we've started with 2071 and then none were removed in our data set. But sometimes you will see that a significant portion are in fact removed. Say, okay, so we've done that. We know that they're all bacteria or archaea. Now let's look at how many sequences are in each sample. Say, okay, so here are the total sequences in each of the samples. And in some cases, you'll see, you know, a huge range of sequences. Like one sample might have like 5,000 and then another sample might have like 120,000. And it can be really difficult to kind of account for the difference in sequences per sample. Especially if you're looking at alpha diversity. So alpha diversity is going to be greatly biased towards like sequences per sample. So you need to be careful with the certain steps that you take when you're doing certain analyses. So let's say there was a sample that had like 1,000 sequences in it while the rest had, you know, 20, 30, 40,000. And say, okay, well, what we're going to do is we're going to go in and we're going to actually just prune samples that have less than or equal to 2,000 sequences, which in our case is going to result in no samples being pruned because they all have more than that. I just wanted to give you an example code if you ever ran into a situation when you're working through your own data set. And so far what we've been looking at is the absolute abundances. So how many times the sequence or the ASV was sequenced per sample, which is great. But sometimes you're also going to want to look at relative abundances. So like, you know, out of the 38,000 sequences from R1-3A, how many of those were, what proportion of those sequences were assigned to, let's say, like Pseudomonas or Firmicutes? Right? So we're going to just go ahead and make a relative abundance phyloseq object by using this transform sample counts command. Now we're ready to start doing some data analysis. We're going to start with alpha diversity. You don't have to start with alpha diversity when you're going through your own data set. It's just what I always like to do. So we're going to start with that. We're going to say, all right, we're going to, I will also say, in some cases, you may want to rarefy your data before you do alpha diversity. Now the community, the microbiome community is pretty split on whether or not you should rarefy and when it makes sense to rarefy. I tend to rarefy if I'm looking at samples with an order of magnitude different of sequences. But that should be something based on your judgment of your own samples. So what I just did with this ordered is I actually ordered the sequence count per sample. So, you know, we're looking at a little over 19,000. And the highest sequences per sample is 48,000. So we're not really talking about an order of magnitude difference here. So I'm not going to rarefy for the purpose of today's tutorial, but just know that it may be something that you want to consider with your own data. So we're going to first calculate some diversity measurements. So we're going to look at Shannon diversity, richness, and then we're going to look at evenness here. So we will run these two lines of code. And don't worry about this error message. It's just because we're only working with 9 samples. It's not something that we need to be worried about right now. We'll calculate evenness, and then we'll say, okay, for downstream analysis, the very easiest thing to do is to combine the alpha diversity measurements into your metadata file. That way, when you like build a plot, it's just pulling from one file instead of pulling from 2 files, which is really important for like ggplot. ggplot really only likes to work from 1 file. So what we'll say, okay, well, we want to paste Shannon diversity into the metadata file as Shannon, okay? We're going to run this line of code, and we're going to do the same for observed, and then the same for, which observed is the number of observed ASVs per sample, which is the exact same thing as richness, all right? Then we're going to say, all right, we want to put evenness in there, and then it does something really weird with the data. So let's take a look at this metadata file. All right, so we have look, we've now put Shannon, observed, and evenness values into this. So what it did was it converted Shannon to a character. So it reads these numbers as a character value instead of as a number. And it did the same for observed, but it did not do it for evenness. So evenness was actually pasted over to the metadata file as a numeric value, which is what we wanted. So what we need to do is we need to say, all right, we need to convert the observed and the Shannon into numeric variables. So we'll run line 273 and 274. And then we should also take a look at some other values to make sure that they're also coded correctly. So typical codes are character strings, factors, numerics, and integers. Things like block, I don't want them coded as a numeric integer. I want it coded as a factor because they're either in block 1, 2, or 3. And so the way to do that is to say, and the same as line, I don't want line coded as a character, I want it coded as a factor because they're from one of 3 lines, right? So what we can do is we can say, all right, we're going to convert those to a factor. So we'll say, all right, block and line are now factors. And now we can start with our modeling. Block, in my case, is a random variable. So what I'm showing you how to do is a linear mixed effects model, okay, which I use the package LME4 here, where we've got the dependent variable, which is going to be the alpha diversity measurement. And I'm going to look at it by line and by flavonoid production. And then I coded my random factor block like this. We're saying you're going to pull all the data from the metadata file. So we'll build the model. And then there's things that there's assumptions that need to be met in order to run like ANOVAs or linear modeling, right? So I gave you some examples of how to test for those assumptions here. A lot of the testing is visual testing of looking at plots and making sure that they fit a certain line or something. The first thing is linearity. So we're going to plot the fitted values versus the residuals. And what we should see is that there's no pattern here. There's plenty of examples online about what a pattern might look like. And what you really want to see is that the dots are evenly spread across the X and Y axis. We'll say, okay, well, the next one that we need to make sure that we're looking at is homogeneity of variance. So this is that each group within line, so we have 3 lines, right? So the variance of Shannon diversity within each of those groups is similar. So we'll test that by using a Levene test. We'll say, all right, let's check it. So it says, okay, well, the Levene's test for homogeneity of variance is, we end up with a p-value of 0.6, which is great. If you see a p-value of less than 0.05, that means that the assumption of homogeneity of variances has not been met. Then we need to look at the distribution of the residuals. And what you want to see is that the blue dots somewhat travel along the black line, which is more or less the case for our data set here. I've worked with a lot of data sets, where I have to transform the dependent variable because there's a lot of issues with the homogeneity of variances and the distribution of residuals. And this actually, this distribution plot looks pretty good. So assuming that all the assumptions are met and satisfied, we can run the ANOVA on the model. And you should note here that this is a type 3 analysis of variance because we have an interaction term, okay? So it has a bunch of information here about the ANOVA and about the linear, the model. And you can see too that none of our variables have a significant effect on Shannon diversity. let's say you wanted to plot it anyways, just because your boss or your professor wants to see the Shannon values across the lines. We can say, okay, we'll plot it with ggplot, where we see that line 321 is the orangish coral color here. And then line 334 is the green and line 336 is the blue. And then we've got the Shannon values on the x-axis. And then we can do this again, all over again for evenness. So I'm actually going to change this to evenness here. And for each of these models, you need to check the assumptions, right? So you'll run the Levine test, you'll plot the residuals versus the fitted values, and then you'll plot the, you'll do the QQ math plot. And then assuming that everything looks good, you run the ANOVA, and then you can plot it. Again, so now we've got evenness values here. And then let's just say that you wanted to plot like the phenolics, right? Even though the phenolics, again, weren't as significant like covariate here, let's just say that we're still interested. Here's two different ways that we can plot it. We can plot it where like we've got all the samples here, so 9 samples are represented by the black line, where we've got the total phenolics on the x-axis and richness on the y-axis, or we can separate it. So we can separate the phenolics and the richness by the actual line, like so. So we can say, okay, for the blue line is the observed and the phenolics content for the sorghum line N336 and so forth. This is kind of a plot that you would want to do if you have a significant interaction between a factor and then a covariate numerical variable. And let's say the ANOVA was significant, and we actually want to look at the evenness model by line. Well, we can say, all right, we're going to look at the estimated marginal means. And we want to do a pairwise, so we want to look at like line N321 by N334, and then line N321 by N336, and so forth. So what we have is the confidence intervals here for each of the lines for evenness values. And then here are our contrasts. So we've got N321 versus N334 and a p-value of 0.397. And then if our covariate was also, like if there was an interaction between our categorical variable or our factor variable and then our covariate, then we can run EM trends and then EMMIP and look at those results. And basically we built, a confidence interval here again, and then we have the pairwise comparisons, which show no significant difference, but we already knew that because the ANOVA wasn't significant. And then let's just run the plot for that interaction, which would be, again, coding or coloring by line, and then the x-axis is total phenolics, and then we've got the linear prediction here across the y-axis. That's just some of the sort of things and ways that you can analyze alpha diversity. These aren't the only ways, but this is obviously depending on what sort of data you're working with as well, right? We're going to move on to making heat maps. heat maps can be very visually easy to interpret, and something really good to include in a paper. Sometimes, though, they don't really add any useful information. But we're going to make some anyways, and I'll explain sort of how to interpret what they're telling us. The first thing that we need to do is we need to run this taxa level function. Now this function comes from the Microbiome Seek package. And I did not have you install the Microbiome Seek package because it is really difficult to install. I've had a lot of problems in the data analysis working group workshops getting it installed, and it's still under beta testing. But this taxa level works really well, and I use it all the time. So like in my papers that I'm going to be using it in, I will cite Microbiome Seek. So if you use it as well, please remember to do that, to cite the Microbiome Seek package. Let's run the function. And then what this function allows us to do is it allows us to take all of the sequence information at a given taxonomic level and just kind of pull it into that level. So what it does is like, okay, we're going to take our phyloseq object and we're going to say, okay, we want to look at the phylum level and the abundances of each phylum. What we can do is we can say, okay, we're going to do that, and then we can plot it here with the heat map function. Let's zoom in and we can say, all right, so we have all of our phylum along the y-axis and our samples along our x-axis. And there, I told it that what I want to do is use the phyloseq object to make a peak away plot using the Bray-Curtis dissimilarity matrix. And I want to do this for each line, right? So I want to name them by the lines instead of it being like R1-3A. I want it to be N33, whatever. So the brightest blue are the highest abundance. So like proteobacteria is very high abundant across all our samples. And the black is least abundant. So we can see here that like SAR, clade, marine group is very, very low in abundance, except it's got a little tiny abundance in this last sample, N334. Okay. A lot of people ask, okay, well, why aren't the lines grouped together? You see here that N334 is right next to N336, and then N336, N321, you know, and then on the other side, we've got N334 again. So why isn't it like all the N321s next to each other and then all the 336s, so forth? It's because we told it to use the Bray-Curtis dissimilarity, which means that the closer together two samples are, the more similar they are. All right, so this N334 on the far left is most similar to this N336 right next to it. And it is least similar to the actual other N334s that are on the right side of the graph. Now you can force it to group them together, but then you lose the sort of dissimilarity aspect that is built into the heat map itself. And then we can do the same at the genus level. But it's almost like there's just too many genera to really be able to read the heat map very well. But I just wanted to give you an example of, you know, what that might look like. And it basically told it, okay, if it's not present, if it's got a 0 as the abundance, then just color it as gray. So there's a lot of different ways that you can change the functions just a little bit to make them more a more visually appealing heat map. Then we'll move on to beta diversity. So there's the main way to look at beta diversity is through a permanova, a permutational ANOVA. But the permanovas have a assumption that there's a the group dispersions are homogeneous. So there's a similar dispersion around the mean for each of the groups that you're looking at, which is not always the case, especially in microbiome studies. So the first thing that you actually need to do before you do a permanova is you need to look at the group dispersions. And you can do that by running this beta disper line of code, right? So you can use beta disper, and then you'll use Adonis for the permanova. And for both of those, for both the Adonis and the beta disper, you'll need a distance matrix. Okay. The most common distance matrix used for microbiome studies is Bray-Curtis. I think that is slowly changing to account for centering or normalizing ASV counts, like doing some like centered log ratio, but I'm not going to show you how to do that today. I'm going to show you how to do this using Bray-Curtis. So we're going to say, okay, we're going to make a couple different distance matrices. The first one is going to be a Bray-Curtis distance matrix of the ASVs. And then we're going to do Bray-Curtis of the phyla. And then since we made a phylogenetic tree, we can do unifrac, right? And I'm going to do a weighted unifrac measurement here, and I'm just going to call it unifrac. And this is at the ASV level, all right? And then it just says, you know, you didn't specify a root, so then we're just going to randomly assign it as ASV 1383. No big deal. And then we can run beta dispersion here, and we're going to do it by line because that is the group that we're most interested in. So let's say, all right, let's do the dispersion. We can look at the ANOVA and say, all right, so the ANOVA, the p-value is 0.8, which is going to suggest that, in fact, there is similar dispersion within the 3 lines that we're looking at, which is great. And we can say, all right, let's look at it. And that was at the phylum level. Okay. We can say, all right, let's look at the phylogenetic, weighted phylogenetic distances using unifrac. Okay, similar. P-value of 0.7, we're good. They have a similar dispersion across the lines. I'll say, let's look at the ASVs and say, okay. And then, oh yeah, we can look at the actual beta dispersion. Like this disp.asv, this is the outcome of beta dispersion, and it shows you some information here. So it says the average distance to the median for each of the lines. So the average distance to the median for line N321 is 0.31. And then we see 0.34 or 0.35 for N334, and then 0.28 for N336. So we can actually like look at those distances. Some people graph those distances because dispersion within their groups is significant to their research projects. So I just wanted to show you where you can actually find those distances. They're included in the output from beta dispersion. Okay. And we can run the ANOVA on that. And the p-value is above 0.05, but it's close to 0.05. So I chose this one so that we can just dig a little deeper. So if it is less than 0.05, you know, here are 2 things that you might want to do. You could do a post hoc analysis using a Tukey HSD, which produces some confidence intervals and spits out some p-values for you. So it says, all right, so when we compare line N336 and line N334, we see that there is a p-value less than 0.05 here. And when we can go back and look at the distances to the median, and that's really not surprising because it's these two comparisons. So, N334 has a greater dispersion than N336. But since our initial ANOVA wasn't significant, we wouldn't have run the post hoc tests, but I just wanted to, again, give you some examples of what to do. And if you wanted to plot it, the dispersions, you could do this. Where again we see that N334 has a greater dispersion than N336 does. But ultimately what we saw was that the dispersions were similar enough across our groups that we can move on and interpret our permanovas more, or have a better interpretation of our permanovas. We can say, all right, we'll look at the phyla level Adonis, and we say, okay, we want to look at line by total phenolics, and there's no significant differences. There just isn't a lot of differences between line, right? So it's a significant factor, but it just doesn't, there's no differences in the phyla level abundances between the lines. We say, okay, let's look at the ASV level, and we're just doing it by line here, and we see, okay, well, again, there's no differences in ASV community composition between the lines, but line has an R squared of 0.26, which is, again, fairly high, especially for a field study, which is where this data came from. And we can look at, yeah, again, I just wanted to show you what you would do if, in fact, the permanova was significant. You would run the post hoc test, which is pairwise permanova, with the number of permutations set. Typically, it's set to 999. I've seen other people set it to 99. And we see between the pairwise comparisons that, in fact, the ASV composition is similar across each of the lines. And then we can look at the phylogenetic distances as well. And it's a similar story here where there's no differences in the phylogenetic composition across the lines. But again, you can run the pairwise permanova if, in fact, the Adonis showed up as significant. The next thing that I think is probably good for me to show you how to do is the bar charts. A lot of people are always interested in just visually seeing a bar chart of, genus level distribution or phyla level distribution or what have you. And the best way to do this is to use the relative abundances because it will make the most sense when you're looking at the final product. And I'll show you what I mean by that in just a minute. So we're going to say, okay, well, we want to look at the phyla level within our relative abundance phyloseq object. And then we want to identify the top 10 most abundant phylum. And then we want to actually just extract those phylums so that we can plot those. And then we use this nifty function, ps_melt, which is going to make the phyloseq object into something that ggplot will read easily. Oops. So then we say run ps_melt. And then I have this colorblind palette here that has 10 colors, and since we're looking at the top 10, then this colorblind palette will fit well for our data. We run the palette, and then we can easily just make a bar plot. And what we did was we said, okay, there's 3 reps for each of the lines, and we're just going to add those together, right? So we added all the actinobacteria together across the 3 lines, and we plotted that for N321. And then we did that for each of the phyla. So what we end up with is a y-axis that goes from 0 to 3, which intuitively doesn't make that much sense. Because what is 3? You know, what is 3 equal here? It would make a lot more sense if instead of adding actinobacteria together from the 3 replications, it would make a lot more sense if we just took the mean of actinobacteria across those 3 replications. So we can do that by running this aggregate function, where we're going to aggregate the abundance of our phyla. I know it says O2 here, but just bear with me. It means phyla for this case, across our lines, right? And we're going to do that based on the mean. Okay. So then we run this, and then we can make a new ggplot. And now look, now what we have is we've got the abundance, and we actually have the relative abundance, the mean relative abundance of that's actually proteobacteria, the bright pink here. Proteobacteria, the mean abundance for each of the lines, and then the mean abundance of Bacteroidea, cyanobacteria, etc. And then we can do this again for like at any level we want to. We can do it for class, order, family, you know, you can do it at the ASV level, but I'm just going to show you how to do it again at the genus level here. There we go. So we've got the top 10 most abundant genera and their mean abundances for each of the lines. And then we can say, okay, well, based on this, it really looks like maybe like Pseudomonas is more abundant in 2 of the lines compared to line N321. And we can easily do like a quick test for that. So we can say, okay, well, we want to look at just Pseudomonas. So we're going to extract Pseudomonas from this file, the meltgen file, which is something that we made up here on line 442. And we'll just call it Pseud because that's easy. And then let's just run like a quick little linear model here. We'll say the abundance of Pseudomonas by line. And in fact, we see that there is a difference in Pseudomonas abundance by line, and then we can run the estimated marginal means again here to actually look at the contrasts. And we see that line 321 has a lower abundance of Pseudomonas compared to line 334. Okay. Something else that I get asked often is how to really incorporate continuous variables into these microbiome studies. And there's several ways to do it. You can do like a distance-based redundancy analysis or a canonical correspondence analysis where you have a nice ordination plot and then you have like vectors of, ours would be like total flavonoids or something. Or if you have like copper or sand composition or something like that, right? I'm showing you how to do it based on, or to do an analysis with that ancillary data with a mantle test. And so what we're going to do is we're going to take our phyloseq object and we're just going to extract the ASV table. And then we're going to look at the Bray-Curtis dissimilarity in that ASV table. And then I just chose dppH here as our continuous metadata variable to incorporate based on the Euclidean distance matrix. There's many different distances that you can use. Euclidean is probably a pretty common one. So we'll say, we'll make that distance And then we can say, okay, we want to compare them. So we're going to put, run the mantle test with our ASV distance matrix and our DPPH distance matrix, and then we're going to try to correlate them using Spearman correlation. And we see when we do that, that we get a mantle R statistic of 0.16 and a significance of 0.18. So this means that there's really not a good correlation between the ASV composition and DPPH and differences in DPPH across the samples. And then we can say, okay, well, we'll look at the unifrac distance. So this is the phylogenetic difference. We can say, okay, now we want to incorporate more than just DPPH. We also want to look at flavonoids. So we could do that by just adding flavonoids in here to the function. And then we can run that again here. We can see that the mantle R statistic is pretty low and it does not seem to be significant. Oh, one other thing that you can do, Spearman is not the only method that you can use for this. You can also use the Pearson correlation, and there might be some other methods for correlations as well for you to use. And I will say that so we're only using 9 samples here, but in total I had 40 total samples. And I will say that I did find a significant correlation between ASV abundances and total flavonoid concentrations. And that is included in the publication that resulted from all of the analysis that I had done. One of the more popular approaches, well, it's popular, but it's also like a hot topic, is this differential abundance analysis. There's a lot of different ways to run this sort of analysis. There's ALDEX, ALDEX2, there's EdgeR, there's DSeq, and then the newest method, I think, is corncob. And this is a algorithm that was produced by Amy Willis from the University of Washington. And I really like this specific algorithm because intuitively it makes sense to me when I plot things. Like I can make sense why something came up as more abundant in one group versus another. And that's really what this is. It's comparing the abundance of a certain group, like at a taxonomic level, between 2 factor levels, right? So we've got 3 lines. So we need to run corncob 3 times. So we'll run corncob on comparing line 321 and 334, and then comparing 321 with 336, and then comparing 334 with 336. And this is the formula. So we've got, or the function differential test. And then we're going to be looking at line, and then I also wanted to incorporate block because block is also a really important thing for us to incorporate as a random variable. And so for us, we also want to put block here for the formula null. So this is going to actually allow us to control for block as a random variable. And we're going to say, okay, we're going to use this phyloseq object for P321 versus 334. And we're using a wall test with an FDR cutoff. So this is a p-value cutoff of 0.05, an adjusted p-value. So FDR is a way of adjusting a p-value. So run this. And then after this runs, we will plot it. So I should also mention that corn cob, while we're just using it here for differential abundance, you can also use it for differential variation. So I don't know if you've ever heard of the word dysbiosis, but basically it's the idea that in one group you may have a similar dispersion of a given taxa. So like Pseudomonas, you may see that, Pseudomonas in one group ranges from 5 to 20. But then in another group, Pseudomonas range from 2 to 200, right? So then the second group would have a differential variation in Pseudomonas compared to the first group that only range from 5 to 20. And this can take a little while to run because it's looking at each taxa, and it's comparing the abundance of each taxa by the lines, by block, right? So it's a pretty complicated analysis that it's running. So we'll do the plot, but let's just go ahead and start running the second differential test here too, while we actually look at the plot. So let's zoom in here at the plot, and we see that ASV2 and ASV9 are higher in abundance in one of the lines, and then there's a lot more ASVs here that are more abundant in the other line. One of my biggest frustrations with these analyses is that it doesn't automatically label like which line should be like over on the left, right? So is this line 321 that has all of these more abundant, or is this line 334? And then, or is this on the right side, are these line 321 or are these line 334? It's one of my biggest frustrations with doing these analysis, which obviously does not sound like it should be that big of a frustration. And then, again, you can kind of just like look at each of these. You'd run this 3 times, and then you'd kind of look at the plots and figure out which ASV is more abundant and which line and so forth. So we can just say, let's choose ASV 9 here to look at, this Pseudomonas. So we'll say we want to, the easiest way to decide whether ASV 9 is more abundant in N321 or N334 is to just do a quick and dirty little plot where we look at ASV9 from the ASV table and we plot it by line here. So say, okay, well, obviously ASV9 is more abundant in N334 compared to N321. Sometimes it's not so obvious, or you may actually want to extract the mean. So we would say, okay, well, we're gonna aggregate the abundance of ASV9 by line, and we're gonna do that by the mean. So we'll just run this line here. We'll say, okay, great. So we've got the mean abundance of ASV9 and N334 is a little over 2,000. And then the mean abundance in line 321 is 209. Quite a bit of difference here in the abundance of ASV9 across these lines. And while I just showed you how to do this at the ASV level, you could also do this at any of the other taxonomic levels. So you could do this at the genus level, class, order, family, phyla level. And that is all that I have for the workshop for you today. I know I ran through things fairly quickly, but I'm also kind of hoping that you'll be able to run through this tutorial before we get to the boardroom sessions, or at least try to run through a portion of the tutorial. That way, if you have issues or questions while you're going through it yourself, we can easily address those at either the Wednesday or the Friday session. If that is not the case, and you aren't able to get to the workshop before those sessions and you have questions for me while you're going through this tutorial, please email me. I'm constantly fielding questions about Amplicon sequencing stuff from colleagues, and would be happy to answer any questions that you're having or problems, try to address any problems that you have. Again, my email address is muc345@psu.edu. I believe that I put my address on my GitHub. No, I did not. I will put my address on the GitHub too. So if you can just find your way to the GitHub, which maybe I'll show you what that address is again, then my email address will be there for you if you should need it. I hope you all have a great time going through the workshop and going through presentations and attending any live sessions. And I hope to connect with you in the future at other Tri-Society meetings or at any other like microbiome meetings and so forth. Thank you all again for registering and for going through this, and I hope that you found this helpful.